In objectives 23 through 32, we'll look at amino acids, which are the building blocks of proteins, and then the proteins themselves. Recall that earlier we saw amino acids as the monomers and proteins as the polymers that are made when we join together amino acids by dehydration synthesis, forming peptide bonds. An amino acid has three parts. There's an amino group. When it's uncharged, that's NH2. When it's charged or ionized, when it becomes a cation, it's NH3+. It's taken on an extra proton. There's a carboxyl group. When uncharged, it's COOH. When charged, when it becomes an anion, because the hydrogen ion, the proton, has left, then it's COO minus when it's charged up. And then in between that, sandwiched in the middle, is an R group. And the R group is important for the function and the properties of each of these amino acids. And when we string together the amino acids into a protein, then the R groups are what are going to give that protein its specific properties. We looked earlier at peptide bonds that are formed in a dehydration synthesis, giving off water. That's what gives the dehydration synthesis its name. So in this case, we have a hydroxyl and a hydrogen, and they're taken off and used to form water, and that forms what's called a peptide bond between a carbon and a nitrogen, as shown here. Another example of a peptide bond is shown in this slide. We have amino acid 1 and amino acid 2. These are joining together in a dehydration synthesis to form a dipeptide. If we repeat this over and over again and form a peptide bond between each of the amino acids, then we eventually end up with a polypeptide, and if we make that even longer, then we end up with a protein. So 2 amino acids and 1 peptide bond is a dipeptide. When we join 2 amino acids together, we do that with a peptide bond. So 2 amino acids are a dipeptide, 3 amino acids are a tripeptide. When we put together lots of amino acids, the Latin prefix for lots of something is poly, so we call that a polypeptide. And then when the polypeptides get larger and larger, we tend to call them proteins. Now, there's no firm dividing point between something we're going to call a polypeptide and something we're going to call a protein, but proteins can be very big, and polypeptides tend to be on the smallest side. So if it's smaller, we tend to call it a polypeptide. If we got, say, a dozen amino acids, we'd probably call that a polypeptide. If we have 3,000 amino acids, then we definitely call that a protein. All 20 of the amino acids have 1 amino end and 1 carboxyl end. The only way that the 20 amino acids that we typically find in the human body differ is in their R groups. And as I said earlier, the R groups are what are going to give these individual amino acids their properties that we're going to study in just a minute, but they're also going to give the proteins, which are made from stringing these amino acids together, their properties as well. And as we'll see, proteins are able to do a whole bunch of different things. They're able to do a whole bunch of different things because of the combination of these different R groups. So we see some examples of R groups here. They can be very, very simple, simply a hydrogen atom in the case of the amino acid glycine, or they can be very complicated, such as a ring structure or a long chain of CH2s capped with an amino group on the end of it in the case of lysine. Now we're going to look at these 20 amino acids, and as we look at these, I want you to remember that I don't want you in this course to learn the structures or even the names of all of them, but I do want you to be able to recognize when I show you the structure of an amino acid what category it fits into. So on the exam, what I'm going to do is show you the structure of the amino acid, and I'm going to ask you to give me the category that it fits into. As we work with these more and more, we'll become familiar with some of these amino acids later on in the course. So the categories that we need to put them into are polar, which, as I said earlier, we use interchangeably with hydrophilic, versus nonpolar, which I said earlier we use interchangeably with hydrophobic. So either polar, hydrophilic, or nonpolar hydrophobic amino acids. The polar amino acids or the hydrophilic amino acids are further subdivided into simply polar uncharged, or polar acidic, or polar basic. Before we look at the amino acids that fit neatly into groups, let's look at some oddball amino acids. The first of these is the simplest amino acid, and so because it's so simple, it doesn't really fit easily into either the polar or the nonpolar group. So that would be glycine, the simplest amino acid that has an R group, shown here in red. It's simply a hydrogen atom. Another unusual amino acid that we use for special purposes is cysteine. When it forms a disulfide bond with another cysteine, then we call it cysteine. So you'll hear biochemists talking kind of funny, and they'll say cysteine, and when they say cysteine, they mean one of these. When they say cysteine, they mean two of those that have made a disulfide bond. Recall that sulfur has very similar chemical properties to oxygen, and so this sulfhydryl group, SH, shown here, has very similar properties to a hydroxyl group. Just like hydroxyl groups can steal a hydrogen atom and form ester linkages, this is something called a thioester when this forms. The sulfhydryl groups can form thioester linkages, which is just a sulfur bridging between two different carbons, just like an oxygen bridges between two different carbons in an ester linkage. When we need to put a kink into a protein chain, then we use a kinky amino acid. The kinky amino acid is called proline, and because of the unique structure of its side chain that loops back around and grabs onto the amino group, then it's able to put a little twist or a kink into the structure of proteins. So acidic amino acids are those which donate a hydrogen ion, a proton. Basic amino acids are those which donate a hydroxyl group and take a hydrogen ion or proton and develop a positive charge as a result. Aspartic acid and glutamic acid are examples of polar charged amino acids that are acidic. So we're going to put those in the acidic category. Lysine, arginine, and histidine. Notice the positively charged groups which have donated a hydroxyl in each case are examples of basic amino acids that have acted as hydroxyl donors. Now, we have amino acids that will hang out with water, so they're hydrophilic, but they don't necessarily donate a hydrogen ion or a hydroxyl ion. Then we call these polar uncharged amino acids. So serine, threonine, glutamine, asparagine, and tyrosine are examples of these polar uncharged amino acids. Tyrosine is a special case because it's very important in the thyroid for making thyroid hormone. Recall that thyroid hormone has a lot of iodine in it. We talked about that when we talked about the functions of iodine in Module 2, and we'll review those again in this module. And so iodine is added on by removing the hydroxyl group from the left-hand side of this molecule and replacing it with an iodine atom. This can happen either 3 or 4 times. If it happens 3 times, then we call the resulting hormone triiodothyronine. If it happens 4 times, then we call it tetraiodothyronine. Finally, we have nonpolar amino acids that are hydrophobic. Remember, we use the terms nonpolar and hydrophobic interchangeably. These are amino acids that don't like to be around water very much, but they do like to hang out with each other. So we want to put together a protein in such a way that we have different side chains with our groups hanging out together and excluding water, then we're going to use hydrophobic amino acids or nonpolar amino acids in that part of the protein structure. The one of these that's really notable is methionine. Methionine, which is abbreviated MET or M, is always the first amino acid in every protein as it's made. Sometimes it's taken off, but every protein begins with methionine. We'll see that in Module 5.